In the last class, we did the sewage treatment and microbes in human welfare. We finished that portion, correct? If I'm not wrong, let me just check. Oh, we have, did we finish the BT cotton part? The BT cotton, etc.
No, sir, I don't think so.
I've not taught you microbes as biocontrol agents, right? Have I? No.
Yes, sir. You have not taught.
Okay, okay. So anyone has any issues with the sewage treatment thing, microbes in the sewage treatment? Because this is one thing which I have seen multiple times in school exams and also in boards. Anyone has any issues? If not, then someone would like to give me a quick summary in 5 minutes. How about Donna? Donna, you were there in the last class?
Yes, sir.
Yes. Would you like to give a quick summary for me, please? Just try, like I'll be here. Yeah, great.
So just
Good,
a
let's
minute.
begin. Yes, sure. Whenever you're ready, Donna.
Yes, sir.
Yep.
Okay. So
Yeah.
we'll first talked about the different microbes, I believe.
Yeah, I think we will start from here. We are talking about microbes only in this chapter. This chapter is microbes in human welfare. So, but we were talking about bioactive molecules and enzymes that we can use in medicine. So I think we started with streptokinase, right? Which is an enzyme. Yeah. So let's begin.
It comes from the bacterium Streptococcus, and it is used as or it helps in like clot buster.
Yeah, it is used as clot buster. You're right.
Yes, and it is used for the myocardinal infraction.
Yeah, myocardial.
Sorry, sir.
Myocardial. It's A.
Oh, cardial. Okay.
Cardinal changes the whole meaning. Cardial means heart, cardiac arrest, cardiovascular system.
What does cardinal mean?
Cardinal is, I think, it's a very respective term used for Christian clergies. Cardinals. It's a group of, if I'm not wrong, like we still have to go and check, because I heard the word in this one beautiful movie called The Two Popes. If anyone has seen The Two Popes.
I haven't.
You haven't? Okay. I'll recommend that to everyone. You know, just between two, it's based on the story of Pope Francis, how he became the Pope, how he is the first non-European Pope in many years, and it's just a talk. The whole movie is about two popes talking. Yeah, you should watch it. It's good. It's not just to believe in the political or any other beliefs that they personal beliefs that they have against or for things, but just to see the beauty of cinema. It's amazingly directed. So I think it's not cardial and cardinal are very, very different terms. So just keep in mind that the word cardio in biology is associated with heart. Okay, cardiovascular system, cardiac arrest, myocardia, etc. Okay.
Okay.
Yes, continue.
So it is used as a clot buster for removing clots from the blood vessels of patients who have undergone the myocardial infarction, which leads to heart attack.
Clots, if not treated, can lead to myocardial infarction. So that's why they are given to patients who have high risk of myocardial infarction or who have undergone a myocardial infarction. Basically, this is heart attack. If you do not treat the clot and there is myocardial infarction in the heart, then heart will suddenly stop working at a moment, and that's the attack. People die instantaneously sometimes due to heart attack. Yeah.
Then another bioactive molecule is cyclosporin A. It is used as an immunosuppressive agent in organ transplant patients.
Yes.
And it is produced by the fungus Trichoderma polysporum.
Polysporum, yes.
And another one is statins.
Statins, yes.
Statins. It is produced by the yeast Monachus purpureus.
Monachus purpureus, yes.
Which have been commercialized as a blood cholesterol lowering agents.
Yes, perfect.
Then we went to microbes and sewage treatment. We started with what sewage was. It was something that majorly consists human excreta and waste in general. And our sewage is undergone sewage treatment plants to clean out the waste. There's 2 stages of treatment.
Yeah, but you missed one point. Why it's important to do sewage treatment is because sewage is something which is lots and lots of water, correct? Because all the human excreta and the toilet waste that humans generate in the cities can only be drawn or cleaned through water. So sewage is basically water only. Yes. And it's fresh water in a way. The source is a fresh water. It's not salty water or water from the oceans. So it needs to be dumped somewhere, correct? We cannot just vanish all that water. So millions and millions of tons of water needs to be dumped somewhere, and we can only dump it in the rivers or the nearby lakes or something around us.
But it needs to be clean if it should be dumped.
Exactly. So that's where this whole headache of cleaning the sewage. First, we generate the sewage, then we clean the sewage because it's important. Otherwise, it cannot be sustainable. We cannot keep getting water and keep producing sewage. Yeah. Yes.
Then it has 2 stages of treatment. The primary treatment, and it basically involves the physical removal of particles, large and small, from the sewage through filtration and sedimentation. Then the soil and small pebbles are removed by sedimentation, and all the solids that settle form a primary sludge, and a supernatant forms the effluent.
Yes.
The effluent from the primary settling tank is taken for secondary treatment.
Yes.
And this is the second part of the treatment. It's also called a biological treatment.
Yes.
The primary effluent is passed into a large aeration tank where it is constantly agitated mechanically and the air is pumped into it. And this allows the growth of aerobic microbes.
Aerobic bacteria majorly. You're correct that there might be growth of fungus as well. Some fungal growths also are seen as long filamentous fungal growths, but majorly it's done for aerobic bacteria because
Does that also help with the treatment?
That helps in creating flocks, in producing the flocks. Because a flock has to be large mesh-like network at the end of the this aeration process, so that after that we also need flocks to be removed, correct? So this aerobic bacteria that grows, it serves 2 purposes. Aeration serves 2 purposes. One, when you start giving oxygen, the anaerobic bacteria dies because it cannot tolerate oxygen. For them, oxygen is toxic.
Yes.
So they die. and aerobic bacteria starts growing up because oxygen is available. And the good thing that aerobic bacteria do is that they consume the organic matter by oxidizing it, because they can use oxygen now. So over the time, the organic matter load decreases. By organic matter, I mean the human waste, urea, nitrogenous waste, ammonia, etc. Okay, so yes, fungal growth also helps, but even if it is not that significant, it's okay.
Okay.
Yep.
And oh yeah, these aerobic bacteria, they consume the organic matter in the effluent, and this decreases in organic matter load. And then this significantly reduces the BOD, which is the biochemical oxygen demand of the effluent. It basically refers to the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were to be oxidized by bacteria.
Yes, correct.
Then it is treated till the biochemical oxygen demand is reduced
To
then,
safe levels. Yeah, when
okay,
BOD is safe, then we just release that secondary effluent into the river, and then we have to worry about the flock that we generated. Yeah.
The flock is the okay. Oh, yeah. The effluent is passed into a settling tank where the bacterial flocks are allowed to sediment. The sediment is called activated sludge, and a small part of the activated sludge is pumped back into the aeration tank to serve as a inoculum. The remaining major part of the sludge is pumped into large tanks that is called anaerobic sludge digesters. And here other kinds of bacteria which grow anaerobically digest the bacteria and then the fungi in the sludge.
So we are doing exactly the opposite, So we generated lots of aerobic bacteria. Now we want to get rid of them. So, but that sludge, that flock part, which is the activated sludge, we can take some of that sludge as an inoculum, and we can put it back into this first here in this first step where I have shown the yes, and again aerobic bacteria can be grown, but the rest of it, almost most of it, we don't want. Want to get rid of it because anyways it will be generated again. So we now start growing the same anaerobic bacteria, like the similar kind of bacteria that are anaerobic. So we cut down the oxygen, we stop the aeration, we cut down the oxygen, and we start growing anaerobic bacteria. So anaerobic bacteria over time, because they cannot oxidize substances and get energy from them, so they start killing the aerobic bacteria and digesting them up. Okay.
Yes.
Now, one question I asked, I think we have discussed it. In this step, we are creating lots of anaerobic bacteria. What happens to this? This NCERT is not told, but you should know, because if you know the logic and science, this question is very logical. So we are killing aerobic bacteria because we generated a load of it. Now, if we allow anaerobic bacteria to grow in the last step, we are again generating a load of it. So what to do with anaerobic bacteria? So it's very simple. You don't have to do anything to anaerobic bacteria. Just start aeration again, and they will automatically die. Correct?
Yes.
Make sense?
Yeah.
Yeah. Okay. Then what happens when anaerobic bacteria kill the aerobic bacteria or the fungi?
It kills the bacteria and the fungi in the sludge. And during this digestion, the bacteria produce a mixture of gases. It contains, I think, methane, hydrogen sulfide, and carbon dioxide.
Yes.
And these gases form biogas that can be used as source energy.
As a fuel source, right?
Yes.
Very well. Great. And if you go to NCERT, all of you have NCERT with you? There's one dedicated portion where they talk about microbes in the production of biogas. So it's the same thing, but it's not like all the time. Biogas is a requirement. We can produce biogas from the sewage treatment plants and also independently of it. So if you want to produce biogas independently of the sewage treatment plants, it needs some biological waste, because there are bacteria called methanogens. They are called methanogens because they release methane when they digest organic matter. Basically, when they grow anaerobically on starch or cellulose, they produce lots and lots of methane. And biogas can be created from cattle's dung, cow dung, or buffalo's dung. So in rural areas where animal husbandry is a very major thing, and people do livestocking or animal husbandry a lot, so they are also producing animal waste, right? Cow dung or buffalo dung, because they are culturing these animals. So there it's easy to set up a biogas plant using cow dung. So what happens there is, it's simple. You just have to make 3 chambers. One is where you mix the dung with water, and then you start sending it into the other bigger chamber, which is called the digester. There, that dung and the water is allowed to mix with methanogenic bacteria. The methanogenic bacteria will start eating up all the organic cellulosic waste in that dung and start creating methane, CO2, H2S, etc. That will be collected. So like there'll be a chamber floating. Yes, Donna, what's your question?
Sorry to interrupt, sir.
No, it's fine.
Did we learn about the Ganga action plan and the Yamuna action?
The Ganga action plan. No, I have not taught you about the Ganga action plan, but it's basically you don't have to learn about the plan. You just have to know. You just have to know that the Ganga action and the Yamuna action plan are created, and a lot of sewage treatment plants have been set up, especially in the areas which are located at the banks of these rivers, so cities and towns. Because earlier, because these rivers were so big, so in the beginning, when something is very big, huge, and you just dump a city's waste in it, you will not feel that the river is getting polluted, right? But
Yes.
over a decade, you will start feeling, oh, now the flora and the fauna of that river, the ecosystem is dying, and there is lots and lots of waste which you can see. So when animals will die in that river, their dead bodies will also float ashore, right? And yes, and that will create more mess. So under these plans, it was proposed to build large number of sewage treatment plants all along the banks. Every town, for example, like places like Allahabad, which is situated on the bank, let's say, okay, or like Delhi is at the bank of Yamuna, or Agra, let's say. So these cities need to have large sewage treatment plants, and it was mandatory for them to only discharge the water in the rivers after treating it for sewage. Okay.
Okay.
So that's all you have to know about Ganga action plan and Yamuna action plan.
Okay.
It's not just for Ganga and Yamuna. It's basically now have been started for all the major rivers in India and worldwide. So that's all, because NCERT is an Indian textbook, so it talks about the Indian plans, but it's logical, right?
Yes.
Every big city situated at the bank of a river needs to treat its sewage before dumping it into the river. So that's all about it. Okay.
Okay, sir.
And
Thank you.
about the biogas production, you can come to your NCERT's page number 186, which is just again a revision. So here we don't have to deal with water or sewage. We are taking dung, which is animal waste, which is less toxic than human waste. We'll mix it with water and we'll allow methanogens to grow into it in a big digester. When they will grow, they will produce methane and other gases in the biogas, and that will be collected by a chamber, and that gas can, as it is, be used as a fuel. And after the digestion is done, then you let the fluid, let that whole solution flow to a third chamber, which is called sludge. There the sludge is allowed to settle, and then you can decompose by just burying it under the ground. It can act as a manure, okay, if something is left. And the water, again, you can just doing the same thing, doing the aerobic and the anaerobic treatment, you can just throw it in the river. Okay, sometimes that whole sludgy water is used by farmers to just sprinkle on the farm in between crops. That can also be done. Cool. Okay, so anyone has any doubts, or shall I begin? So the thing I'm going to talk about today is more of a genetical thing, but it's 26 today. I'm going to talk about microbes use as biocontrol agents. Now, what do we understand by the word biocontrol? What do you think biocontrol means? Control means to keep something in check and balance, right? Bio means life. So clearly you are controlling something which is living, and we are using microbes to control some other living substances. So basically what the NCERT or what the heading wants to say is that you can use microbes as insecticides and pesticides without using chemicals, because insecticides and pesticides are chemical agents, right? They are chemicals that have been developed to kill unwanted pests and insects that kind of destroy the crops, right? But instead, if you can use some microbes to control some other microbes, that will be great, right? Now, do you understand? First of all, why we use insecticides and pesticides? What is the need of it? Because we want to get, yes, Donna.
So that the insects don't like destroy the crops.
Exactly, because we want to get maximum yield possible out of our lands, right? Because we have more mouths to feed than we can produce. That's what they say. So we have to maximize the yield. One way to maximize the yield is to reduce the loss by the attack of insects that feed on the same crop that we feed on. So we don't want them to be there. For example, there are many beetles that destroy many commercial crops like cotton. Now, if we want to increase cotton production, we have to grow healthy cotton plants. Now the problem is we can do it by insecticides and pesticides, and in case of cotton it's fine because we don't eat cotton. But in case of other edible crops, those insecticides and pesticides are harmful chemicals which also over time harm the humans, the consumers, correct? That's why they say that you should clean, wash your fruits, vegetables, and your other edible substances should also be washed and thoroughly cleaned before cooking. But still some traces of these chemicals will go inside us. We cannot avoid it. But what we have now started doing is that in agriculture, first thing that we need for the farmer and also in general for the ecosystem is we just cannot keep killing things generally. Like if I put an insecticide or pesticide, it's a harmful chemical, it's like a poison. So it will kill those insects also that are harmful to us, or which are causing harm to us by destroying our crops. But in general, it will also kill other kind of organisms, right? Birds for that matter, or something else. So we want to maintain these complex systems under checks and balances. We also want to maintain the vibrant ecosystem, but we also want to increase the yield, and we want to get rid of the pests specifically. Now, to do this specifically, not just in agriculture, but in general, many microbe-based practices have started. For example, to control mosquito population, what do we do? What do we do in general? Forget about microbes. If you want to control mosquito population somewhere, we cannot make mosquitoes extinct, correct? Why? Because they are very important part of the ecosystem. Yes, they transmit and they act as vectors for many disease causing pathogens, and they cause a harm to human and human life. But we cannot just take revenge by killing all the mosquitoes and making them extinct from the planet, because they are also very crucial pollinators for small grass families. So we will lose lots and lots of grasslands if there are no mosquitoes, bees, and butterflies. So they have to be there in the ecosystem. But we still have to keep mosquito population in control in urban areas, also in rural settlements. But how do we do it? We use chemicals, right? We burn these mosquito killing substances. We use all these chemical substances. We use DDT spray. Okay, right? All these kills the mosquito larva. But instead of apart from this, one thing that can also be done is to use dragonflies. So dragonflies and ladybirds, they are very useful to get rid of mosquitoes, and also aphids. Aphids are small insects which are also very troublesome to humans. So if you purposefully grow dragonfly population, so dragonflies do not cause harm to us. They will just be, and they predate on mosquitoes. So if mosquito population is somewhere proliferating beyond the control, check and balance, introducing some dragonflies in that region will help to keep them under check and balance. So to control mosquito population, dragonflies and ladybirds, these are beetles. They are called ladybirds. These are small beetles with a head and a body and markings like this. I'm sure you all must have seen it, polka dot markings, red and black. Have you seen these
Yes.
small beautiful beetles?
Yes, sir.
They are poisonous, by the way. And those red and black dots are warnings in nature that don't eat me. Okay, many birds do understand the warning and do not predate on ladybirds. Okay, so this is one way to use a living system to control, keep check and balance over other living system. But here we are talking about microbes. So clearly dragonflies and ladybirds are not microbes. But one thing, so we will come to bacteria. So there is one bacteria called Bacillus thuringiensis. Okay. Now, if the name is Bacillus, what does it mean? It means that it is rod shaped. Bacilli, right? And it's annotated as, it's abbreviated as BT. Bacillus thuringiensis is called BT. This is where the word BT in BT cotton has come from. Now, Bacillus thuringiensis can be used to control caterpillars, okay, of insects. Now, how they work, what they do is, so basically, if insects eat, or in the larvae of the insects, that insect lay eggs, and sometimes by insecticides we try to kill them in the stage of the eggs itself. But most of the time when they become larvae, so this bacteria, if it is present in the surrounding, and the larvae, while they are eating that leaf, so larvae has just one thing, one goal and ambition in life. Any insect larva, what does it do? Larvae do not undergo reproductive phases, they do not reproduce, they just do one thing. What do they do?
Eat leaves.
They eat, and keep eating. So they just simply eat, sleep, repeat on record. Because insects undergo this stage called metamorphosis, where they have to make a cocoon around themselves, or it's called pupa, and they have to get into it and transform from a larva into a winged butterfly or a winged insect. All insects have to go through this phase known as metamorphosis. So for that metamorphosis stage, for days, they don't eat. They are inside a pupa transforming. They need lots and lots of energy, because all their organs are recreated from the scratch. New organs are created that larva did not have, like wings, eyes, antenna, legs, etc. They all need to come up from the scratch. So it needs lots and lots of energy. So that's why a pupa just a larva just eats and grow in size, become very fat, and then pupate, go under a cocoon. So they keep eating the leaves, and if on these leaves this bacteria is present, then this bacteria in the gut of insects start producing a toxin. Okay, and that toxin, what it does, it makes small holes in the gut of the larvae. It perforates the gut. Now gut perforation in a larva is a lethal thing. Larva cannot survive because now everything. So imagine that in the intestine there are small holes that these bacteria's toxins create. So they die at the larval stage. Now purpose solved, right? If they die at the larval stage, done. They cannot grow, become the insect, or they cannot keep eating the leaves. Correct? So one way is what people started doing is that they started spraying these bacteria. So these bacteria come, you can easily get it from the market as dried spores. You know, bacterial spores. Under unfavorable and dry conditions, bacteria undergo a spore-like stage, which is called bacterial cyst also. So this bacteria spore or bacterial cyst is like an inactive stage where they still are living. They are not dead as per, but they are inactive. So the moment they will find suitable condition, they will start replicating, dividing, and become active. So you can get these as powder, sachet, dried spores of these bacteria. When you mix it with water and spray onto the plants, then they become living bacteria. And then if these larvae, the insects come and lay their eggs on the plant, then the larvae eat the bacteria, the larvae die. So that's one way to use microbes as biocontrol agents. Is it clear, everyone? Is it clear?
Yes, sir.
Yeah. Now, this way we can also make sure that only that insect will die whose larva will try to eat the plant that we want to grow. Correct? Other insects which are not harming our plant but are just coming and sitting, maybe helping it to pollinate, or maybe just were tired and they wanted to take some rest. They sat on our plant, started tasting something here and there, but because if there is chemical on it, then they will die. Correct? If it's a poison thing that we spray, then everything that comes in contact with that plant dies. So this way we can just restrict to kill specific species which is acting as a pest and not the other insects. Is it clear, everyone?
Yes.
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 So you can write down Bacillus thuringiensis, Bacillus thuringiensis, are sprayed on plants and also on fruit trees. We also do it for fruit trees, and fruit trees where they are eaten by the insect larvae, where they are eaten by the insect larvae, full stop. The BT, which is the Bacillus thuringiensis, the BT produces toxin in the gut of larvae. To kill the larvae, or the larvae gets killed, whatever. This is where we now are using a microbe as a biocontrol agent. But it's again like, getting bacteria from the market, making it into an active form, from a cyst form, then spraying it over the plants. Instead of this, what if now, the next chapter that I'm going to start, which is biotechnology, I'm just giving you a glimpse of trailer of it here only. So now scientists, what they have done is they have genetically encoded, so this Bacillus thuringiensis, whatever toxin it produces, so that toxin must be a gene, because toxin is a protein, so the bacteria must be having a gene for that toxin, correct? Yes or no, people?
Yes, sir.
So if we can take out that gene for that toxin from this bacteria and can make a hybrid plant, a genetically modified plant that we have done for cotton, so that cotton is called Bt cotton. So it's a cotton plant, and each and every cell of the plant will contain the gene and the protein, which is the toxin. So now, without the bacteria, we don't have to spray the bacteria also now. We just know that this toxin will kill only this specific type of insect which eats, which is a pest for this plant. So the insects that are pest for cotton plant, when they will now come and eat the cotton plant leaves or the stem or whatever part of it, this protein which is already present in the plant will get into their guts and will do the same thing, make the guts perforated, and they will die. So is this a better way? No need to spray the bacteria even. Okay. So these kind of plants are called genetically modified plants, and we have created one such through using the toxin gene. The toxin gene we take and we genetically incorporate. Now, how do we do that? The whole next chapter is all about this only. So you can keep your questions for the next chapter, which we'll start today only. So how do we do this? But we genetically incorporate the gene into plant, and this gives rise to genetically modified plants, GMPs. Now we can genetically modify any organism for that matter, but there is a big ethical constraint. You have to get license and permission from authorities before doing these kind of experiments, because we cannot keep making genetically modified organisms and leaving them in wild. We have to be 100% sure that they are not going to destroy the ecosystem. Okay, so in case of Bt cotton, in many parts of India, Bt cotton has been grown, and in the last decade, it has been a big success. And some states of India have actually taken up, and they cultivate Bt cotton on a large scale. So genetically modified plants. If it is GMC, it is genetically modified crops. If it is GMO, it is genetically modified organisms. Okay. So one example is Bt cotton. There is Bt brinjal also. That there was a big debate about it, because people accepted Bt cotton, but many naturalists and many other social people did not accept Bt brinjal as such, because this is something that we are going to eat, right? And cotton is okay, we don't eat cotton anyways, but about brinjal we have to be more careful. But it's not cultivated in many places, but it was one of the achievements of this technology. So Bt cotton, Bt brinjal, both these plants have been successfully made using this genetic recombinant technology. Is it clear, everyone?
Yes, sir.
So,
Yeah. Okay, very well. Cool. So this is about, there is one more there's one more species apart from Bacillus thuringiensis. I'm forgetting the species name, but it comes under the genus Trichoderma. Trichoderma you have also studied in the last chapter, microbes, disease, human health and disease. So Trichoderma, some species of Trichoderma are also used to get rid of several plant pathogens. Okay, so you can just write Trichoderma are very effective biocontrol agents. They are very effective biocontrol agents. Trichoderma is a fungi, okay? Against several plant pathogens. So some pathogens specifically cause disease in plants. For example, some kind of tumor, some kind of wilting of the leaves or rotting of the leaves or rotting of the roots. These happen. Anyone who is interested in gardening and does a terrace gardening or something? No one. If you do that, you must be knowing that there are many kind of plant diseases that happen. So you have to actively keep looking, taking care. It's not like just grow plants and give them water, it's done. You also have to keep looking for certain diseases like yellowing of leaves, wilting of leaves. Sometimes leaves develop holes, sometimes they develop tumors, kind of thing, cysts. So those are caused by plant pathogens. So Trichoderma is a fungi which is very, very effective biocontrol agents against many pathogens. Apart from this, there's one more, it's a virus. It's called polyhedrovirus. I think it's called nucleopolyhedrovirus, commonly known as polyhedrovirus. So I'm writing its name. So you can write down. Viruses. So they also kill. They also specifically kill, I'm so sorry, insects. So insects which affect plants. So they affect arthropods. If you don't want insects around your plants, and the polyhedrovirus belongs to the category of baculovirus. Just know it. Most of the time they will ask you for this term. They're also known as nucleopolyhedrovirus. Again, one more specification. But if you remember polyhedrovirus or baculovirus, it's the same thing. They affect arthropods and they kill the arthropods, and they are also used as biocontrol agents again for many plants. Okay. So basically all these are insecticidal. These are insecticidal approaches or insecticidal what do you say, applications of using microbes, because all we are focusing now till now is to kill the insects. In this also we are killing the larvae of the insect by Bt, which is a bacteria. In this we are using a fungi to kill the several plant pathogens, which can be bacteria and which can also be other microbes. In case of this, we are using a virus, polyhedrovirus, to affect arthropods, again insects, and used as biocontrol agents for plants. We sometimes also want to protect our plants from other higher animals, but for them we do not use these, because when we go high in evolution, it's like we are coming close to humans. So all of these Trichoderma, polyhedrovirus, Bt have been selected to use because they have no negative effect on other plants, mammals like us, birds or fish or any other higher organism. So they are very safe for us. Okay. but they are not safe for insects. Is it clear, everyone?
Yes.
Okay.
Yes.
Cool. So this was microbes as biocontrol agents, which is an easy part. And one last thing in this chapter is you can use microbes as fertilizers. So I should say, because they are living system, I should say biofertilizers. And this, I think you all know. You have some idea of using microbes as biofertilizers, right? In your junior classes, you must have read about this thing, mycorrhiza. You know what is mycorrhiza, people? Mycorrhiza. So mycorrhiza is a fungi. It belongs to the fungi community, and it is a very famous fungi which is known to make symbiotic associations with plants. Okay, so what they do is they form associations and they form nodules with plants where this fungi is living on plants or in sometimes in close association with the tissues of the plant. Now, whenever there is a symbiotic relationship or a symbiotic association, there has to be a dual benefit, right? A mutual benefit, something that plant should get from this fungi and something that this fungi should get from the plant. So can you think what is the plant giving the fungi? Of course, the plant is giving the fungi a home, right? And food.
Yes.
Correct. So you are getting food from the plant because plants are primary producers. They can do photosynthesis that fungi cannot do, and they can give starch to the fungi. So fungi can just suck on the starch from the plants and live happily. But there has to be something that fungi has to give to the plant in return. What can fungi do that plants cannot do that great?
Fixing nutrients in the soil.
Just in the soil? You're right about
Nutrients to
the,
the plants.
sorry?
Nutrients to the plants.
Nutrient to the plants, yes. So you're right about the nutrient plant. So nutrient part. So what fungi do, they absorb or they take phosphorus from the soil, which plants are not very good at absorbing. So they absorb phosphorus and pass it to the plant. Okay, so plants having such associations show benefits, and the benefits is that if these fungi are associated with their roots, other pathogens cannot attack the root. So they show resistance to the root pathogens. And because they also get nutrients or minerals, or I should say these elements, important elements like phosphorus, these plants can show tolerance to drought conditions, to dehydration, or to salinity, all. So overall, it makes it's like a plant become more stronger and show better development and growth, better resistance against pathogens, better tolerance against salts, against less water, which is drought, and against pathogens. Yes. Is it clear? So you can write down mycorrhiza form symbiotic association. So do you think phosphorus, if you know what are fertilizers, one thing which is very famous about fertilizers, we remember NPK. Most of the phosphorus provide most of the fertilizers provide NPK. What is NPK?
Nitrogen, phosphorus, potassium.
Yes. Okay. Great. Nitrogen, phosphorus, and potassium. So phosphorus is provided by fertilizers. Can you see that the same phosphorus here is being provided by this fungi? So that's why it is acting as a biofertilizer. Correct? Microbes acting as fertilizers. They're making the plant more fertile, also more tolerant, healthy, and developing. So form symbiotic relationship. And with the rice. Mycorrhiza shows so this mycorrhiza is the symbiotic relationship. So mycorrhiza contain two things, plant and fungi, and the fungi which makes this mycorrhiza is it mainly belongs to the genus, if I'm not wrong, it's Glomus. Just remember this genus. Fungi from the genus Glomus makes the mycorrhiza, and they form symbiotic relationship with plants. And second thing, they provide phosphorus to plants. And where do they get phosphorus from? They absorb it from the soil. It's not like they make phosphorus, they just provide phosphorus to plants, absorbing it from the soil. So that, please write that also. If you think you can forget, then write that. And some additional benefits also write down. In addition, the additional benefits are plants become more resistant to root pathogens. Apart from the fungi, as for biofertilizers, how many of you have heard about cyanobacteria? Cyanobacteria. What are cyanobacteria? The word cyano comes from the color, okay, the cyan color. They are basically bacteria, okay. They have one more name which is slightly misleading. You must have heard of this name, I'm sure. Have you heard of the name blue-green algae?
Yes.
Yes.
So
Yes, sir.
blue-green algae are not algae, they are bacteria. So just write down aka, also known as blue-green algae. They give an appearance of an algae because when the cyanobacteria grow, they grow like a film, and people in paddy fields especially, cyanobacteria play a very important role. You know what are paddy fields? Like the rice cultivation. In rice cultivation, it is one of the most important biofertilizers. So blue-green algae, which is a cyanobacteria, it adds to the organic matter and increases the fertility of rice plants. So what they can basically do is they can fix atmospheric nitrogen into the soil or in the water in which they are living around the plants. Some examples that you must have done in junior classes is Anabaena and nostoc. So Anabaena and nostoc are some of the blue-green algae, which basically are bacteria, and they act as biofertilizers, act as important biofertilizers. For paddy plantation. Okay, because they are providing, and what are they providing? Mycorrhiza was providing phosphorus, they are providing nitrogen. Fix nitrogen from atmosphere to soil. And then plants can take it from the soil in the form of, so nitrogen is always fixed in the form of nitrates or nitrites that plant can absorb. Plant cannot absorb nitrogen, or they cannot take direct nitrogen from atmosphere. So again, fertilizers have nitrogen, phosphorus, and potassium as components. So if something is providing this, it's acting as a fertilizer. Here, organisms are providing this. That's why they are acting as biofertilizers. Is it clear, everyone? So these biofertilizers are actually commercially available. So if I ask you what is the benefit of using biofertilizers, again, the same kind of benefit that was using microbes as biocontrol agents over insecticides and pesticides is that it reduces the dependency on chemicals. So we no longer have to use chemicals, or we have to use less chemicals. So chemical fertilizers are not very good. Everything clear, people? So I think with this, it marks the end of what we have to cover in this chapter. So no doubts, right? I'll stop here for 2 minutes. If you have any doubts, by the time I'm changing the notebooks page to biotechnology. Yes, you can see this screen now, everyone.
Yes, sir.
Yeah. Okay, now I give you a glimpse of what we can do through biotechnology while teaching BT Cotton. So first, let's come to the word. So I'm starting with the new chapter, biotechnology. In this chapter, we will be focusing on the principles, the concepts, and the processes. Applications is a whole new another chapter, but it's not possible to teach just the principles without teaching you the processes. If I'm teaching you the process, I'm also going to tell you about the applications, but then applications in detail, some of it we will do in the next chapter. But just remember that I will take both chapter 11 and 12 together. Okay, so some things that I'll teach, and you will, if you cannot find it in your chapter 11, don't worry, it must be in chapter 12 where it's application of biotechnology. But basically it will be to build your concepts. Okay, but majorly I will also focus on principles and processes. Okay, so what does the word biotechnology to you means? There are 3 words that I can dissect out: bio, techno, and logy. Let's go to word. Bio means
Life.
life, or can I say living system, right?
Yes.
Logy means because this we have already done, that's why. Logy means
Study.
study. Techno means simple, it means techniques, right? Which basically a logical process is called a technique. Correct? So biotechnology, what do you understand? If we club it together and try to make a definition of our own, let's first make a definition of our own, then let's see how it fits with the definition of the book. So what do you think is biotechnology? There's a living system. Yes, sorry, please continue.
Living with techniques, something like
Living with techniques, then where does this study comes from?
living with technical studies.
Living with technical studies, that sounds like an engineer's life, living with technical studies. Oh my god, how can I bear this? This is how to live. Something more elaborative. Living with technical studies sound like living with our sorrows to me. I don't know why. It's like living with this pain of studying biology to become a doctor. So it's a field that deals with, so biotechnology, if you know, just from your career perspective, anyone who's interested in engineering and also have a thing towards biology and who wants to do both the things, there's something that is called a bioengineer. You can become a bioengineer. I must be thinking, what will I be making if I'm a bioengineer? Of course, not bridges and buildings and towers and skyscrapers. You will be engineering biology. Biotechnology, if you have heard, there is a BTech degree in biotechnology. You know that? And there's also a BSc in biotechnology.
Yes.
Yeah. So you can be an engineer through biotechnology, or you can be a non-engineer and also do biotechnology. So what it means is, it is a field that deals with study of techniques. Okay, so we develop techniques, scientists develop techniques where they use living systems or living organisms or component of living organisms to produce products or services for the humankind. Make sense? So it's the
Sir, can you
study.
repeat?
Yes, I will. So let's write down. Biotechnology is an integration, is an integration of living systems and their components with techniques to produce with techniques to produce with, you can also write with techniques of natural science, with techniques of natural sciences to produce products and services. Okay, if you find this hard to remember, in very simple words, you can also say that biotechnology deals with study and development of techniques of using living system and its components. And by components, I mean enzymes, proteins, etc. to produce products and services. Whichever you find easier. This is more compact and concise, but technically heavy. This is simple and elaborative. Anything which you can remember better. Both of these are definitions for biotechnology. Is the field clear? Is it clear what we are going to do now? What we are going to study? This part is clear, everyone? Yes?
Yes, sir.
Perfect. Now let me tell you biotechnological applications. So you have studied till now without knowing that what we are studying comes under biotechnology. A lot of applications from biotechnology. In the chapter reproduction, I told you about something called IVF, right? You all know what is IVF? What is IVF? In vitro fertilization. What is in vitro fertilization? We use two components of living system, which are gametes, male gamete and female gamete, take them out of the organism's body under controlled environment and developed, artificially created environment that is again created by engineers, these bioengineers. we make these gametes fertilize in a test tube, and then we implant it back into the mother's body. So it's a biotechnological application, okay, to assist in the reproduction of couples who cannot conceive naturally. So IVF is clear. Anything else that comes to your mind? Very recently, we all have been talking about
BT.
before, even something more common than that, vaccines,
Oh.
especially the RNA and DNA vaccines. So all the vaccines that you take for COVID, like your Covishield, Pfizer, Sputnik, Covaxin, AstraZeneca, they are all RNA vaccines, RNA protein, okay, RNA vaccines majorly. So this also is a biotechnological application that we can use RNA from the virus and can use it to develop vaccines. So vaccination has been greatly influenced by biotechnological advances. And as you said, again, yes, genetically modified crops, GMO, for example, genetically modified organisms, for example, BT cotton. Again, we have been able to do it through biotechnology. Anything else that comes to your mind that again is an application and we have already studied?
DNA fingerprinting.
DNA fingerprinting. Yes, kind of. DNA fingerprinting again, but what? Yes, we are creating a service there, which is identification of parents. Correct? DNA fingerprinting. We are using a component of a living system, which is DNA, and creating a service for it. Anything else? There's something called gene therapy. Have you heard of this term, gene therapy? For many rare diseases, gene therapy can be done. You can use some gene, knock it out, express it, overexpress it, underexpress it, and can cure a condition or a disease or a disorder. That's again an application of biotechnology. And there are many more. It's like using gene or synthesizing something in a bacteria. For example, one more application is gene synthesis. Gene synthesis and gene expression. So suppose we want to produce some hormone or some enzyme for human use. Let's take an example of insulin. You know, when we first discovered that insulin can be used to treat diabetic condition, and the first dose of insulin was administered, where was that insulin taken out from? We cannot take out insulin from human and give it to human, right? It's
From a cat, cow, something like that.
no, it was taken out from pigs, the pig pancreas. And pigs have a great potential because first they are mammals. They are close to us in evolution, and so are like rats are also mammals, but they are small. So we can use rats as model organisms or mouse as model organism to do drug testing or to do fundamental science, to test for certain receptors, things like that. But in pigs, because it's very good size as well, it's a very potential animal to do mixed genetics. So I don't know if you are aware, but recently in US, in the early this year, there was a interesting news that came out that a human who needed a heart transplantation and who was like terminally, he was in a terminal condition, he was about to die. So a genetically modified human heart, which was raised inside a pig, was transplanted to this person. And this person survived for around 2 months on a heart, which is a genetically modified heart of a human raised and grown inside a pig's body. So this is a very promising start. So that person died after 2 months due to immune complications and infections of that transplantation itself. And things like that. But this is a very promising start for us to understand, okay, larger mammals can be used to harvest human organs. Now, there are many ethical and social issues associated with it that science will deal eventually as it will grow. But this is one scientific feat of scientific achievement, correct? But it's a very big thing. Even simple things that we normally do in our life, like insulin injections. Every diabetic patient takes insulin injection, right? When required. Have you ever thought, where is that insulin coming from? From the microorganisms. We can synthesize insulin in yeast, single yeast cells or bacterial cells, just by not taking our gene and putting it inside the bacteria or the yeast. Okay, so they are expressing our genes, making that protein for us, and then we purify that protein and use it for therapy of humans or treatment of humans. So all these comes under biotechnological applications. Okay, is it clear, everyone? These are some of the examples, not all. Anyone has to ask something? No. Okay, so now let's first go to understand the principles. Every field, or for example, even engineering, medicine. In medicine, there are many fields, like some doctors are cardiologists, some are gynecologists. But in every field, even if you are into any other profession for that matter, there are certain principles by which that field grows and develops. Okay, so there are some principles of biotechnology as well, and to be very precise, there are just two basic core principles of biotechnology on which you can build layers and layers and make it as complex as you want, develop it as much as you want. But the basic principle of biotechnology is that there are just two core techniques that enabled the growth of this whole field. So what are those two core techniques? And those techniques also tells us about what this field majorly does. So whatever we do through biotechnology, either we are doing genetic engineering, which means we are engineering the genetic material. This is what a bioengineer does. So genetic engineering. Okay. If you can say, these are engineers of living systems, engineers of life. So genetic engineering means you take a DNA or a RNA, which are genetic material of a cell, and you alter the chemistry of it. You modify, you do whatever desired changes you want to do in that gene, and then you put back that gene into the host organism and see what changes now will happen when you changed the genetic makeup. You engineered the genetic material and you put it back into the organism. Now, what phenotypical changes that host organism shows? That's called genetic engineering. Is it clear, people?
Yes,
Yeah.
sir.
Yeah.
Yes.
So this is the first core technique. So this is what biotechnology is based on. You have to make modifications to the chemistry of DNA or RNA. So in simple words, write down genetic engineering. It includes techniques. It includes techniques to modify the chemical makeup, the chemical composition of genetic material. Now, genetic material can be DNA or RNA. and introduce it into the host organism to study the phenotype of the host organism. Okay, so that's called genetic engineering. Simply engineering the genetic material of an organism. So this is one core technique. The other core technique is which is called, if you are not engineering the genetic material itself, then what are you engineering? You are engineering the conditions. You are engineering where you should do that. For example, let's take an example of IVF. The brilliancy or the magic of IVF, scientific magic, is not that logically we can make two gametes fertilize each other outside the body. But the achievement is that we manage to create an environment outside the body, because if you take anything outside the body, for example, blood or any tissue or these gametes or our cells, and keep it on any surface, they will die, right? They won't survive. There is a reason why sperms survive in the spermatic cord or in the testes or in the male system, is because they get the optimum condition. There's a reason why fertilization happens inside the female, because they get optimum chemical cues and conditions. So to be able to develop the similar conditions, same simulations outside the system, a biological system, is also a biotechnological achievement, and that we call as bioprocess engineering. So either you are engineering the genetic material itself, or you are engineering the process. Bioprocess engineering. Is it clear, everyone? Am I making sense, or am I just blabbering some?
So does inventing like the materials required for this also come under this bioprocess engineering?
Yes, inventing or creating sterile contamination-free ambience, creating these very fine micro needles by which we can, if you are making, we can pick one ovum or pick one sperm and do a, you must have read of intracytoplasmic gamete injection, right? So inside the cytoplasm of the ovum, we can inject a sperm. Now imagine a sperm which is, we cannot see it with the naked eye, but you can pick it up with a fine needle. Now that needle is something that we made, right? We did not get it from the nature. The environment in which we are doing it, to make the buffer or a chemical solution which exactly simulates the chemical environment which is present inside the female system. And to be able to do this, even if you tweak the phosphorus, calcium, any of these concentrations, the sperm will die. If the environment is slightly acidic or slightly basic, the sperm will die. If the environment is non-sterile, there is a single bacteria present, the sperm will get contaminated. Okay, so to be able to make and maintain sterile ambience in any chemical engineering process, okay, to be able to regulate which organisms we want to grow and which organisms we do not want to grow, to be able to make products like antibiotics, vaccines, enzymes, they come under bioprocess engineering. Make sense, people?
Yes, sir.
Yeah.
Yes,
So
sir.
yeah, so write down. Bioprocess engineering includes creation and maintenance, creating and maintenance of microbial contamination free sterile ambience. microbial contamination free, or we can also call it sterile ambience in chemical engineering process. Chemical engineering processes is the genetic engineering is also a chemical engineering process. Must have heard of engineers who are called chemical engineers, right? Ever thought of what does a chemical engineer builds? They build chemicals. But they're engineers because they are engineering something from the scratch, structurally also. So these chemicals have a structure. Okay, antibiotics, enzymes, vaccines, they all have a structure. So chemical engineering process to regulate the growth of desired microbes in large quantities. To regulate the growth of desired microbes in large quantities. So both these two core techniques are clear to you. Whatever we are going to study now in this chapter will belong to either of these one core techniques. So either we will be learning about principles and processes of genetic engineering, or we will be learning about principles and processes of bioprocess engineering. Okay, is that clear?
Yes, sir.
Yeah. Okay, so I think we don't have time left now. We have 1 minute. So I'll stop here and I'll stop for questions. If you have questions. And in the meanwhile, please write this homework that you have to do, because now the class is going to be very technically heavy. I'll try to break it, grind it as much as I can to explain you in the least technologically heavy terminologies, but some things we cannot avoid. For example, so your homework is you have to go and familiarize yourself with certain words. You know, go to internet sources, try to understand, or you can go to NCERT also if you find it there. You have to figure out what is a recombinant. What do I mean by recombinant DNA technology? So this is what we are going to study the next in this chapter. And plasmids. Plasmids you have studied in your junior classes, that they are extra chromosomal DNA fragments present in a bacteria. But try to get revise it and try to find a little bit more about it. And third thing I want you to read is about gene cloning. What do we mean by cloning a gene, which is very obvious by the word itself, and gene transfer. And just do it for bacteria for now, prokaryotes. Okay, so this is your homework, everyone. Please do this. It will help you to understand the next class better. Otherwise, you'll be blank a little bit.
Sir?
Yes, Lubna, you have a question?
Yes, sir. So
Yeah.
you said about this the person who had heart transplant, and like it was from
Yeah.
like the human heart was like inside the pig, like genetically it was grown inside a pig. How did they do?
It was a genetically modified heart, so it was not a human heart, because
So it
the
was
shape
not
of a
exactly a human heart, just
yeah, it was a hybrid of a human and a pig heart, somewhere in between. Okay. So it was done because, see, it's very obvious. No, if I take a human heart and put it in a human, there are going to be complications, right? Because even from human to human, that the organ is not 100% accepted by the body, because anyways it's a foreign tissue. Howsoever it is coming from the same species, it is a foreign tissue. It's genetically not identical to us. The most what do you say, seamless transplantation is between two twins, identical twins. If monozygotic twin, one twin gives something to the other, there still will be complications, but not that much. Body can easily accept it comparatively. But here, if I take a heart from a pig and put it in a human, the human should die the next day. a pig heart should not work in a human. We are two very distinct, different species in evolution. We do not share a common ancestor like anywhere soon near in the evolution. So the whole experiment was to see if we genetically modify, we make some some component of human genetics also in a heart of a pig. So now that pig's heart, can it be accepted by a human body to some extent? And to some extent it did. Like the human survived for 2 months, which is a big leap for science. But then ultimately he died, which everyone they knew that anyways he's going to die, but he got 2 months extra life and also served for science. So yeah, that's all. It was not a pure